Welcome to another screencast where we'll be discussing the mechanism of how DNA replicates itself, or DNA replication. So before we start this, you need to be extremely familiar with the structure of the DNA molecule. And if you're not, then you need to do that. Otherwise, this is going to make a little bit less sense. So there's some things you need to know, especially regarding the whole 3' 5' stuff. So we're going to be able to hopefully describe and explain the semiconservative model of DNA replication, and then to be able to detail the specific roles of the following in how the leading and the lagging strands are replicated. So we have several enzymes, DNA polymerases, helicase, single-stranded binding proteins are involved in this. There's an enzyme called ligase. There's another enzyme, RNA primase. RNA primer is not an enzyme, but will play a role. And then there's an enzyme, telomerase, as well. So let's get started. And when Watson and Crick elucidated the double helix structure, they commented in their paper that a copying mechanism or replicating mechanism became apparent to them. And if DNA was going to be the hereditary material, then it had to have a way, the cell had to have a way to copy that and pass it on from one generation to the next. So this molecule hinted at that, the structure of it hinted to them how that might happen. And you might take either side of that molecule, right, break these hydrogen bonds as you see here, separate them, and then use each side as a template, right, to then add the complementary side to that. So you use this as a template, and then you add the complementary nucleotide there. And you do the same on this side over here and add the complementary. And in that way, you're able to get two exact replicas of the exact same sequence. So if you notice ACTAG, ACTAG, the exact same there. So you're going to get two identical nucleotide sequences there. So how does that happen? Me describing it is one thing. I think that one of the best things you can do is actually go to these animations that I've posted the links to on Blackboard and to watch it over and over and over again. Okay, but I can describe it to you here, and then you can go watch it. So the semi-conservative model, and there's another screencast that discusses the experimental evidence that led to led to scientists concluding that it was the semi-conservative model and it was not this conservative or dispersive model, all right? So the semi-conservative model is what we just described, where the parent model opens up and each side of the molecule is used as a template to then synthesize the new daughter strand, right? So here we go again, the parent DNA molecule at the top of this slide, right? The replication begins at origins of replication, right? And so we see these several origins of replication all along the length of the chromosome, and this is where the molecule will open up. And so what we see, you can actually see this on electron micrographs, is this bubble, right, where the DNA is separating from one another. The parent strands are separating, and you get these bubbles, and they're referred to as replication bubbles. So these bubbles are several along the length of the chromosome. And at the end of each bubble then is an area known as the replication fork, right, because it's forking right there. So they call this the replication fork. So this, then as the replication continues, right, these arrows are the direction in which the replication is happening. Right? So we're unzipping the molecule this way, unzipping it this way, and then adding new nucleotides that are complementary to the parent strand. Right? And in such a way, then you end up getting two daughter DNA molecules down here that are identical in sequence to the original parent molecule up here. But you see that this daughter DNA molecule here is half of that. The one side is the original, and then the other side is new, right? And then new, and then original. So one thing to test to see if you understand this, can you describe the relationship between these two? Right, so that would be relationship A. And then what's the relationship between these two, B? And then what would the relationship be between these two, C? And then the relationship actually between this one and this one would be relationship D. So there's a way to kind of test to see if you understand this and you're able to describe those relationships. Right? So some things you need to know about this replication. Right? First of all, as we discussed in class, you are making a polymer here. So the enzyme that's going to be involved, one of the enzymes is known as DNA polymerase. Right? And so just some things that you need to know, a background about DNA polymerase, right? It will synthesize, right, or make phosphodiester bonds. Phosphodiester bonds between nucleotides. And so we detailed that, that bond in class. And it will only add, it can only add nucleotides to the 3' end of an existing nucleotide. Now, this definition here at first seems a little wieldy and you may not understand what's happening, but hopefully in time you will. So you need to remember this one thing, that DNA polymerase here can only add nucleotides to the 3' end of an existing nucleotide. So just remember that for now. Another enzyme that is going to be involved here is an enzyme called RNA primase. Now, RNA primase will add nucleotides. It can add nucleotides de novo, that is from nothing, right? So that does not, this enzyme here does not need a pre-existing nucleotide to start laying down. Okay. Another enzyme that's involved early on in the process here is this helicase. Is a helicase enzyme, and helicase will break the helix open. Right? So this will break the hydrogen bonds between the two sides, or the two chains. Right? And so this helicase is what generates these replication bubbles initially at the origins of replication. Right? So helicase, again, opens up at the origin of replication the parent DNA molecule, making the bubbles, right, the replication bubbles. Moving on. We're going to look at this leading and lagging strand here. We're going to look at this. The box is around the replication fork, so we're going to zoom in and look and see what's going on at that replication fork, but also what's happening here at the origins of replication. Right? So there's two things going on at the exact same time here. Right? There's something going on up here, right, and there's something going on down here. So if you notice, like this side right here, this new strand that's being synthesized looks different than this new strand that's being synthesized down here. So this is different than this. Right? So on either side of the fork, things look different. And on either side of the origin of replication, things look different, right? Like this new strand that's being synthesized here looks different than this new strand that's being synthesized over here. This is because the molecule is anti-parallel. I'm going to try to focus on one side at a time. Let's look at the leading strand first. So at the origin of replication, what happens is the helicase opens it up, and then we have this enzyme primase. Right? So this is primase. This is also the RNA primase I was talking about. So here's the enzyme RNA primase, and it lays down, right, at the origin of replication, or near there anyway, an RNA primer. So this little red piece right here is an RNA primer. And this primer is a little short nucleotide sequence. Right? It's an oligonucleotide sequence. So this oligo is short or few, right? It's the opposite of poly, which would be many. So oligonucleotide sequence, and it's made of RNA nucleotides. This seems strange, I know, but we'll come around to that a little bit later. So first step, helicase opens it up. 2, primase lays down an RNA primer. This RNA primer has a 3' end right there. Right? So now the next thing that's going to happen, right, is that DNA polymerase now, shown down here, can come in, bind to this molecule, and begin to add nucleotides to the 3' end of that RNA primer. And once it gets started, it can then continuously catalyze the reaction, right, in this direction, because here's a 3' end. This RNA polymerase is actually going to move, right, and change its conformation. It changes its shape, and when it does that, it actually moves down the DNA molecule, pulls the DNA molecule through it, so it sort of ratchets its way down that molecule. And each time it does, this 3' end enters the active site here, and it can then catalyze, right, bringing in another nucleotide, right, a free nucleotide here, adding it to that 3' end. So the DNA strand, the new daughter DNA strand, which is complementary to the parent DNA strand, so this is complementary, right? This is complementary to the parent strand. That new strand is synthesized, new strand, synthesized in a 5' to 3' direction. Always, always, always. DNA polymerase synthesizes new strands in a 5' to 3' direction. Right? It's growing, it's extending on this 3' end. Okay? Now, you're trying to make identical DNA copies, right? So, well, the original DNA parent strand or parent molecule didn't have any RNA in it. So what happens is, toward the end of this, another DNA polymerase here, right, a different DNA polymerase but similar, right, you see the shape is different but similar, will come in and actually remove this RNA primer and add DNA in there. Right? We can talk about that detail a little bit more later. But so the DNA polymerase, this is DNA polymerase 3 doing that over here. This is DNA polymerase 1 over here, right, removing the RNA primer. Right? And so now the daughter strand is complete. Right? So let's look at that in terms of what's going on at the fork. Right? Right here. So, confusing diagram, but let's look and see what we know. Okay. Here's our parental DNA in dark blue, 5' end, 3' end. Follow this 5' end here, 5', following it all the way here, and there's a 3' end. What they're not showing you on this diagram is that this would have kept extending, right? And right here, complementary to that on this 5' end would have been the RNA primer. So since that is the 5' end, this would be the 3' end of the primer. DNA polymerase has latched onto this molecule and has now moved down this way. And as soon as you begin, right, the DNA polymerase ends up down here, and so this is the 3' end. Right? So this DNA polymerase is continuously synthesizing new DNA, right, the daughter DNA molecule toward the replication fork. So this DNA polymerase 3 synthesizing toward the replication fork. Right? And it's continuous. Once it starts, it continues to be made. And so this is referred to as the leading strand, the leading strand up here. Incidentally, we'll back up and just point out a couple things in this diagram. Right? This green thing right here is an enzyme. Which enzyme do you think that is? If you said helicase, you are correct. So that's the helicase enzyme. These things right here, these are the SSBPs. Right? This stands for single-stranded binding protein. Single-stranded binding protein. And this single-stranded binding protein holds the parent molecule apart and prevents it from rehydrogen bonding back together again. Okay, so leading strand, start with the helicase, opening it up, RNA primase lays down a primer. That RNA primer then can have DNA polymerase 3 begin to add DNA nucleotides, 5' to 3' direction, continuously toward the replication fork. Now, the problem with this molecule, not the problem, but the issue, is that on the other side, since this is the 3' end, and this is the 5' end of the parent molecule, right? When you open up at the origin of replication, right? This is the 3' end. Sorry, this is the 5' end, and there's no 3' end for the DNA polymerase to begin adding to. So primase has to lay down a primer, right? So if you lay down a primer right here, right? Here's the RNA primer. Here's RNA primase. It lays down a primer. Which end of the molecule is this of that primer? It's the 5' end. Means the 3' end is right here. Okay, no problem. But DNA polymerase is going to add only the 3' end. So here's DNA polymerase 3 adding nucleotides 5' to 3', but the problem is it's moving away from the fork, and so you're left with these, this parent strand not replicated there. As this helicase is kind of moving down this way, this is going to continue to unzip and break. Right? So the this side right here, you have this DNA polymerase 3 replicating away from the fork. Right? Then here's another primer laid down, right? 5' to 3'. DNA polymerase 3 replicated back that way. Here was DNA polymerase 1, removed the primer and converted the primer into our DNA. Right? And then there's another enzyme right here, DNA ligase, that seals up this last little phosphodiester bond between what used to be primer and the daughter strand on that side. Right? So this strand down here is known as the lagging strand because there's a lot more going on and it takes longer to synthesize this strand, so it lags behind the synthesis of this leading strand. Right? Because you have to wait for this to open up here, you have to lay down another primer, right? And then when you lay down another primer, it's synthesized away from the fork. So if you understand what just happened there, you should be able to see these different fragments known as Okazaki fragments, right? And let's number them. Let's call this Okazaki fragment 1, Okazaki fragment 2, Okazaki fragment 3, and then Okazaki fragment number 4, right? So which one is the oldest? Which one is oldest, meaning which one was synthesized first, which one was synthesized second, which one was synthesized third, etc. Right? And again, I think the best way to see this is watch these animations. So if you can, look at the PowerPoint and go to those animations there and watch this all happen, that'll be a little bit more helpful. Right? Problem is at the end of the linear molecules that the DNA polymerase runs out of the end, runs off the end, and we have a little gap that is not going to be replicated. And so each round of replication, each round of cell division, the chromosome, the DNA molecule, actually gets shorter and shorter and shorter, right? Because that's where that primer was, right? So here was our parent strand. It opens up, parent strand, right? Lay down a primer, right? 5', 3', runs this way. DNA polymerase runs, builds a new one, 5' to 3'. Right? Then this primer comes off, and you're left then with this little gap, this little overhang here that doesn't get replicated. So if you did this again, if you were to replicate this chromosome again, the next round of cell division is going to get shorter and shorter and shorter, right, each time. So the way that the cell handles that is it has an enzyme called telomerase here, and that enzyme has built into it an RNA molecule, right? So there's this combination of protein and RNA here. And this RNA molecule is lined up such that there's the 5' end and here's the 3' end. And so this telomerase enzyme gets onto the ends of the chromosomes. Right? And now this is a 3' end for DNA polymerase to begin to add to. Right? And this telomerase sort of moves this way, and you can then add, DNA polymerase will come in, add nucleotides in there, fill in that gap there, and the chromosome remains intact, remains as long as it was when it started. Right? So the recent Nobel Prize in 2009 was awarded for the discovery of this telomerase enzyme. And again, I would recommend watching those animations. Okay. So that is a lot of information, and you want to be able to sort of watch this, listen to this maybe again, watch these animations, and describe the roles that DNA polymerase 1 and 3 play. Right? What does the helicase do? These are single-stranded binding proteins. What is the job of the ligase? Describe the role of RNA polymerase and the role of RNA primers in replication, and then a little bit about the telomerase enzyme. Okay. Thanks for listening, and do the best you can. We'll talk about this in class. Watch those animations.